Hello, thanks for coming to see my talk. My name is Arsham Nejad Kourki, and I'm a final year PhD student at the University of Bristol, School of Biological Sciences. I work as part of the paleobiology research group, and the talk today is part of my PhD project, the evolution of the eumetazoan body plan, and it's titled Support for a stem-eumetazoan Dickinsonia. So just to give a bit of context, as the title suggests, my PhD project is about the evolution of the eumetazoan body plan. The reconstructing early steps in the evolution of eumetazoans, including reconstructing their body plan, has been a difficult thing, even though people have been working on it for literally several decades. So, for example, we're still not quite sure whether cnidarians, which have a sometimes bilateral or quasi-bilateral, sometimes radial body plan, whether this body plan is ancestral to eumetazoans or the bilaterian body plan is ancestral to eumetazoans. We're not sure exactly what are the homology relations between the germ layers of bilaterians and cnidarians. We're not entirely sure how the body plan evolved. We're not entirely sure, whether our mouths are homologous to the mouth of a jellyfish, or is it our anus that is homologous to that, or something in between, or something a bit more complicated. So most of the evidence for this stuff comes from comparative morphology, developmental biology, developmental genetics, more recently comparative genomics. But the only source of direct evidence, namely paleontological evidence, has been kind of difficult to come by. And the reason is that when the eumetazoans, or the early metazoans more generally, were kind of evolving first and radiating, when they originated, was in the Precambrian. And it's quite commonly known that in the Precambrian, in the Ediacaran especially, where we find most of the fossils that we do find, the fossils are mostly not very easy to find. They tend to be quite scarce. And when we do find them, they tend to be very enigmatic. They are scarce because the organisms that lived back then, the ones who were macroscopic, tended to be soft-bodied, unlike Cambrian and later fossils. And they tend to be enigmatic because they're very difficult to make sense of, because they don't tend to have most of the characters that modern animals do have. So this makes using paleontological evidence to better understand the early evolution of eumetazoan characters tricky. So this talk is about Dickinsonia. Dickinsonia is probably the prime metazoan candidate for amongst the Ediacaran fossils. Most of the Ediacaran fossils, we're not really sure whether they're animals or not. Dickinsonia we're a bit more certain about because of its overall morphology, its quasi-bilateral body plan, macroscopic size, and evidence for active locomotion. These are some of the main things, as well as geochemical evidence. So the really, really cool thing about Dickinsonia, came out in the paper a couple of years ago, is that in the fossils of Dickinsonia, there are traces of steroids, so like things like cholesterol, that fit that very strongly fit a metazoan or animal profile. So we're pretty sure that it was an animal. What kind of animal it was, it's harder to say, because it kind of has some features of eumetazoans and lacks some key features of eumetazoans, such as a gastric cavity. So the evidence suggests that it might be a stem-eumetazoan, but in order to be able to more confidently make this kind of claim, we need to do a phylogenetic analysis, obviously. So that's what we've done here. And I'm going to try and take you through some of the main characters of Dickinsonia that we use for our morphology-based phylogenetic analysis. So the fossils that we have of Dickinsonia come in two general forms. We have the body fossils and the trace fossils. So the body fossils are what's left of the impressions left by the body of the organisms in the sandstone. So sand would have come up from above, come down from above and left and the body fossil would have left an impression. The trace fossils would have been left by the organism's traces, feeding traces. And these are preserved in a slightly different way, as we'll see in a couple of slides. And it seems like that the trace fossils were most likely formed by the active removal of the microbial mat covering the seafloor, which was very, very common back in the Ediacaran, but this kind of ecosystem kind of disappeared with the Cambrian explosion. Also, so this probably means that these organisms fed on the microbial mat, much like modern placozoans do today, except those are microscopic. And the orientation and distribution of these trace fossils very strongly suggests that these animals had active directed locomotion. So they didn't just move around randomly, they moved in preferred directions, which, by the way, suggests that they had sensory cells somehow distributed around them, and also that their lower body surface was used for feeding. Furthermore, with the body fossils especially, they reveal a modular quasi-bilateral or bilateral body plan, which most likely resulted from a highly regulated ontogeny. And we can see that from comparing smaller or more juvenile and more mature Dickinsonia specimens. Further evidence, for example, some specific specimens reveal evidence of posterior growth, muscular contraction, and epithelia. So I'm just going to go through a couple of specific fossils to have a look at these things. So this is a body fossil along with two feeding traces. As you can see, the feeding traces have some of the marks of these ridges that you find on the body fossil, but as the feeding traces get older, the marks become fainter, and that's probably the result of the microbial mat growing back as the organism moves to feed on other parts of the microbial mat. And this is a scenario of how it happened. And as you can see on the right, the footprint of the trace fossil is preserved in positive hyperrelief. So it's bulging out of the underside of the sandstone. And the body fossil is preserved in negative hyperrelief, kind of impression on the inside. So this is another really interesting specimen, which bears wrinkles that kind of show that it probably contracted using epithelial muscles. And so using these and a bunch of other key characters that you can see in this table, we infer the position of Dickinsonia and the closely related Yorgia, which has a couple of small differences compared to Dickinsonia. Using these characters and using a Bayesian analysis using method of Bayes, we infer their placement as stem group eumetazoans. So that means Yorgia and Dickinsonia are more closely related to eumetazoans, which would be bilaterians and cnidarians and ctenophores, than they are to placozoans and poriferans and non-metazoans. One thing that's left unclear and unresolved is whether Dickinsonia and Yorgia form a clade, because we didn't have particularly high support at the node joining Dickinsonia and the eumetazoans. So that remains to be resolved. Our analysis was unable to resolve that using the characters we had. And just to mention again, this was a entirely morphology-based analysis, which did take into account some developmental traits as well. So just to wrap up and talk a little bit about implications. So our results showed that Dickinsonia and Yorgia are most likely stem group eumetazoans. We attained these results using paleontological, morphological, and developmental evidence. The monophily or paraphily of Dickinsonia plus Yorgia remains unresolved, which could possibly be resolved by incorporating other Ediacaran organisms such as Charnia, Spriggina, and others not mentioned here. So the main implications, so the implications are quite broad potentially. It can affect our understanding of the evolution of a host of eumetazoan fossils. Eumetazoan, early eumetazoan characters, including the body plan, locomotion, nervous system, etc. And more specifically, and something that's more of a focus of my PhD project, is that these challenge the long-held hypothesis that the radial body plan of cnidarians and ctenophores is ancestral to eumetazoans, and that it evolved out of the evolution of, or rather it emerged out of the evolution of a gastric cavity from a more blastula-like stage in evolution to a more gastrula-like stage, which is called the gastraea hypothesis, after the gastrula stage in development. And rather, it seems like since Dickinsonia and Yorgia are flat, they don't have a digestive cavity, they just use their lower surface to feed, but nonetheless are bilateral. This strongly seems to suggest that bilateral symmetry actually preceded the evolution of gastric cavities. So that should kind of help us make us think about the evolution of the eumetazoan body plan again. And this is something that I am working on as also as part of my PhD project. With that, I could finish up, and thank you for coming. Bye.